These notes are from Chapter 11, Section 3, and they continue with Punnett squares that are beyond Mendel's principles of strict dominant and recessiveness. So the examples that I'm going to cover today involve genetics, but in a different capacity than just one allele is dominant over the other. So we're going to start with incomplete dominance. And incomplete dominance is defined as a situation where neither allele is completely dominant over the other. And a blend between the two traits occurs with the heterozygote. The offspring will be different, completely different from the parents with the heterozygote. And a good example is with snapdragons. Snapdragons are a type of flowering plant. The flowers look like this. And when you cross a red snapdragon with a white snapdragon, and for the sake of simplicity, we're going to use capital and lowercase again, you would expect that the heterozygote would be red, but instead you get a blend where you get pink. And so essentially you're getting some of the trait showing up and creating a new trait. So big R would still be red, little r would be white, but the heterozygote would be pink. So let's just do a couple of examples. If we were to cross a red plant with a white plant, we would do our genotypic and phenotypic percentages. And in this case, we would get 100% big R, little r, and 100% pink. Now the next problem has us doing a pink plant, crossing it with another pink plant. And so we are gonna, again, draw our Punnett square. A pink plant is big R, little r. Another pink plant is big R, little r. We'll fill in our boxes. Remember to bring over and down. Capital letters go first, lowercase letters go second. We're going to do our genotype percents and our phenotype percents. In this case, we've got 25% big R, 50% big R, little r, and 25% little r. Now, in our previous examples of just strict dominance for monohybrid crosses, we would have 75% red, but not this time. We've got 25% red, 50% pink, and 25% white. So again, these types of problems are called incomplete dominance. The key word here is you get a blending of traits with the heterozygote. And that heterozygote is going to look different from both of the parents. The second type of expression is called codominance. And with codominance, both alleles are expressed, but not blended to make something new. Neither allele is dominant or recessive. They are expressed equally with no blending. A good example are, the layman's term is checkered chicken. However, the proper term is called erminette. And erminette chickens look like this. They're absolutely adorable, but you can kind of see why they're called checkered because they almost look like a checkerboard with their black and white feathers. And the alleles that we're going to write are going to be a little bit different. We're going to use the letter C for chicken, or you could use an F for feather. It honestly doesn't matter. And we're going to do a superscript to denote the color of the feather. So we're going to just put a C, and they're all going to just be capital C's. Superscript B is going to represent black feathers. C superscript W is going to be white feathers, and this is listed up above. So in order to have a black feathered chicken, you would write C, B, C, B. A white feathered chicken would be C, W, C, W. And a checkered or erminet chicken is CB, CW. Now you might be wondering, well, why can't I just go BB and WW and leave the C's out? Well, the reason is because you are technically not supposed to change the letter for the same allele or the same trait. So the letter is actually C. The superscript is denoting what version of the allele it is. So that is the proper way of doing it. If you do it this way, you'll probably get the same answer with regards to your genotypic and phenotypic percentages, but the actual documentation of the cross would be technically incorrect. So if we were to cross a black feathered chicken and a white feathered chicken, we'll do our Punnett square. CB, CB, CW, CW. I'm actually listing the alleles in alphabetical order, so I'm putting the B before the W. If we were to do genotype, we would get 100% CB, CW. And if we did phenotype, we would do 100% checkered. Now, if we were to cross a checkered chicken with a black feathered chicken, so an erminet with a black feathered chicken, we would get CB, CW, that's our erminet chicken, and our black feathered chicken would be BB. And here are our results. Again, remember to bring down, bring over. We're going to go in alphabetical order, so the B's are going to go before the W's. And our genotypic and phenotypic percentages would be, for genotype, 50% CB, CB, 50% CB, CW, and for phenotype, we'd have 50% black feathers and 50% checkered feathers. So again, that's called codominance. Both alleles are expressed, not blended. They are expressed equally, and again, no blending. Our last bit here that's slightly different, an extension of our strict dominance or monohybrid crosses, are called multiple alleles. And with multiple alleles, there are more than two versions of the allele. So we've just been doing like tall versus short, or purple flowers versus white flowers. Well, what if there were 3 or more versions of the gene? That's called multiple alleles. And a great example is blood type. There's actually 3 different allele versions out there, A, B, and O for human blood type. Now, you cannot inherit all 3 versions. You can still only inherit 2 alleles for your blood type, one from your mom and one from your dad. But there are 3 different varieties that exist in nature. There's also some rules. So A is dominant to O, B is dominant to O. So if you inherit an A and an O from each of your parents, you're going to have A blood. If you inherit a B and an O from each of your parents, you're going to have type B blood. The only way to have O blood is to get 2 O's. And so O is actually recessive to both A and B. Now you might think to yourself, well, O must be the least common blood type, but actually it is the most common blood type. Of all of the blood types, O is actually, even though it's recessive, the most common. So what about A and B? What if you got an A allele and a B allele? Well, they are codominant, and now you know what that means. They are expressed equally with no blending. So if you inherit an A allele and a B allele, you would be type AB, which is actually the least common blood type. So if you have A blood, you actually have cell surface proteins, marker proteins, if you remember those way back when, first semester when we covered the cell membrane, that essentially are A marker proteins. If you have B blood, you have B markers. If you have AB blood, you have A and B markers. And if you have O, you have no markers. And that is why O is called the universal donor. You can give O blood to other people because there's no markers and the body won't recognize those cells as not belonging to you. So for example, if I have A blood, you can give me type O blood in the hospital if I ever needed a transfusion, because my body won't recognize the O blood as not belonging to me. But if I were given B blood, those B markers do not match my blood type. And also, I make B antibodies. Now that we've talked about COVID, remember, B antibodies are going to tag these B cells and say, these don't belong. So what will happen is your body will try to destroy those blood cells, and actually it can be fatal. So you make B antibodies if you have A blood. You make A antibodies if you have B blood. You don't make any antibodies against blood type if you have AB blood, which is pretty cool because if you have AB blood, you are called the universal receiver. You can receive A blood, B blood, and O blood. And again, the O blood, they're the universal donor, but they have A and B antibodies. So they can only receive O blood if they have O blood. Now, the proper way to write blood type is with an I. And so we write I superscript A or I superscript B, or if it's O blood, we can just do a lowercase I and an O. However, I know that that can be cumbersome. I know it can be cumbersome with the chickens too. So for blood type, I am going to be okay if you just want to write the letters A, B, and O, even though it's technically cheating in a way with writing out your Punnett squares. So in order to solve these problems, we have to read what they say. A male who's heterozygous for type B. Okay, if he's heterozygous, he's not BB. He has type B blood, but he must be BO. And he marries a female who's heterozygous for type A. Well, again, she can't be AA because that would be homozygous. She must be AO. So your cross is going to be BO crossed with AO. And you're going to put the alleles in alphabetical order. So the A goes before B and the B goes before O. And so we have 25% AB, 25% BO, 25% AO, and 25% OO, But for phenotype, we're a little bit more clear about what the blood type is going to be. So 25% AB blood. For this one, it would be 25% B blood. You would not write the O because it's what the trait is, not what the genes are. So BO is actually just going to be type B blood. 25% A blood and 25% O blood. Let's do our last problem here and then we'll be done. So let's cross somebody who has O blood with somebody who has AB blood. If they have O blood, they have 2 O's, and if they have AB blood, they're an A and a B. So AO, AO, BO, BO, geno, and pheno percents. So we've got 50% AO, 50% BO, and our phenotypes are going to be 50% A blood and 50% B blood. So again, this is an example of multiple alleles with a little dash of codominance. Multiple alleles are where you've got more than 2 versions of the allele, 3 or more, and you can still only inherit 2 of the different types because you're still just getting one of the versions from mom and one of the versions from dad. But more than 2 versions exists in nature. And that's it.